Bismillahirrahmanirrahim. Cholesterol. We're going to discuss structure, chemistry, sources, distribution, absorption, transportation, functions, and abnormalities of this major sterol which exists in our body called cholesterol. As regards as structure, all steroids have a cyclopentanoprehydropenentine ring system. It is a fused ring system made up of three cyclohexane rings made up of a three cyclohexane rings designated as ring A, B, and C and cyclopentane ring D. The six-membered rings are in penetrine arrangement. Total 27 carbon exist in cholesterol all derived from a 2 carbon unit compounds acetyl-CoA. One hydroxyl group at position number 3 at carbon number 3 which is characteristics of all sterols. The hydroxy the hydroxy group is beta oriented projecting above the plane of the ring. Double bond exists between carbon number 5 and carbon number 6. 8 carbon side chains beta oriented attached to the carbon number 17. So now we're going to discuss the differences between steroids and sterols. Steroids are often found in association with fats. All the steroids have similar cyclic nucleus resembling penetrine ring A, B, and C to which the cyclopentane ring is attached. It is designated as cyclopentanoprehydropenetrine nucleus. Methyl side chain occurs typically at carbon number 10 and 13. The side chains at position 17 as usual. All the compounds all the compound which contains one or more hydroxyl group they are called sterols. But in steroids no hydroxyl group placed at carbon number 3 or some other carbon. Instead of side chains, steroids have a carbonyl and as well as carboxylic group they are attached. Cholesterol is amphipathic with a polar head hydroxyl group. With a polar head hydroxyl group, they attach with the non-polar cyclopentanoprehydropenetrine ring and hydrocarbon side chains at carbon number 70. Most of the cholesterol in the body exists as a cholesterol esters with a fatty acid attached to the hydroxyl group at carbon number 3. Now the sources of cholesterol. Almost all nucleated cells, including arterial wall, can synthesize cholesterol. The major site of synthesis of cholesterol in liver, adrenal cortex, testes, ovaries, and intestine. Enzymes involved in its synthesis of cholesterol partially located in endoplasmic reticulum and partially in cytoplasm. Sources exogenous sources dietary cholesterol approximately 300 to 400 milligrams per day obtained from a butter, cream, milk, egg yolk and meat, etc. A hen's egg of weight 20 ounce give up to 250 milligrams of cholesterol. Endogenous source Cholesterol synthesized in the body from two carbon unit compounds called acetyl-CoA, approximately 1 gram per day or 1000 milligrams per day cholesterol synthesized. Cholesterol obtained from diet, obtained cholesterol also obtained by from the de novo synthesis in liver, as well as the cholesterol synthesized in extrahepatic tissues and brought out in liver by the help of reverse cholesterol transport mechanism. Now the liver cholesterol pool is formed. So this liver cholesterol pool they further utilized to form uh, vitamin D, to form a bile acids and bile salt, as well as they become the part of the free cholesterol become the part of the bile along with the phospholipids. They also involved to uh, secretion of lipoproteins, VLDL and HDL, etc. Values for cholesterol. Total cholesterol in our body, it should be less than 0.5, less than 5.17 millimoles per liter. That is a good range. 5.17 to 6.19, that is the borderline, and above this, above more than 6.20 millimoles per liter, that is very high level of cholesterol that may be increase the risk factors of cardiovascular diseases included atherosclerosis and hypercholesterolemia. Clinical aspects. Serum cholesterol is correlated with the incidence of atherosclerosis and coronary heart diseases. We know that in case of hypertension, in case of cigarette smoking, more level of LDL, low density lipoproteins, more becomes elevated, more cholesterol becomes elevated, they're going to be oxidized in arterial wall and it leads to a incidence of atherosclerosis. Occurrence are widely present in body tissues. Adult brain and nervous tissues have 2%, liver up to 0.3%, skin 0.3%, intestinal mucosa 0.2%, endocrine glands, adrenal cortex contains 10% or more, corpus luteum also rich in cholesterol. Cholesterol also present in blood and bile and usually a major constituent of a gallstones. Forms of cholesterol. Cholesterol occurs in two forms: free form, equally distributed between plasma and RBCs, and esterified form, also called a bound form, in which the esterified cholesterol esterified with a fatty acid at hydroxyl, hydroxyl group at carbon number 3 position. Fatty acids that esterified with a free cholesterol: linoleic acid 50 to 60%, oleic acid 18%, palmitic acid 11%, arachidonic acid 5%, and other fatty acids 16% esterified with a free cholesterol to make them cholesterol esters. In brain and nervous tissues, free form predominates, whereas in adrenal cortex it occurs mainly in esterified form. Now absorption and transportation. Cholesterol esters, cholesterol which associated with cholesterol esterified with fatty acids, are present in food we eat. Cholesterol esters hydrolyzed by a pancreatic enzyme, cholesterol esterase. Products of enzyme digestion forms micelles with bile acids in the intestinal lumen. Micelles interacts with the enterocyte membrane, allows diffusion of lipid-soluble components across the enterocyte membrane into the cell. The bile acids, however, did not enter the enterocytes at this time. They remain in the intestine, travel further, and are reabsorbed and sent back to the liver by the enterohepatic circulation. This allows the bile acids and bile salts to be used multiple times in fat digestion. Now, the transportation of cholesterol. The intestinal epithelial cells will resynthesize the lipoproteins. The lipoproteins are particles that made by the aggregation of triacylglyceraldehyde, free cholesterol, cholesterol esters, phospholipids, and the specific proteins called apolipoproteins. All these substances aggregate to make a lipoproteins called chylomicron, which originates from the mucosal cells, excreted into lymphatic system, ends up end up in blood circulation where they transport dietary lipids to all the tissues of the body. Now, inhibition by drugs. Cholesterol, endogenous cholesterol which synthesized in the body, so that should be inhibited by statin drugs. Statin drugs included lovastatin, mavastatin, atorvastatin, simvastatin, and fluvastatin. They inhibits the cholesterol biosynthesis by decreasing the activity of HMG-CoA reductase, a regulatory enzyme of cholesterol biosynthesis. Now the functions of cholesterol. Major structural constants of cell membrane and plasma lipoproteins which involved in plasma lipoprotein which found in plasma involved to transport fatty acids, tags and sterols. Cholesterol serve as a precursor for a variety of biologically important products including steroid hormones, progesterone, glucocorticoids, mineralocorticoids, androgens, male sex hormones, female sex hormones, estrogens, as well as bile acids and bile salts that derive from cholesterol. Bile acids and bile salts which derive from cholesterol act as a detergent in the intestine, emulsifying dietary fats to make them readily accessible to digestible enzymes called lipases. Bile acids and bile salts synthesized in liver, stored in gallbladder, and released into intestine after intake a diet rich in lipids. Some of the bile acids and bile salts were named cholic acid, chenodeoxycholic acids, which further conjugated in liver with the amino acid glycine and taurine to make them a taurocholic acid and glycocholic acid and tauro chenodeoxycholic acid and glycokenodeoxycholic acids. They are primary bile salts which synthesize in liver. Assignment: draw the structure of cholesterol, also describe its biochemical function. Enumerate bile salts, also give their function.